everyone! So our topic for today is all about DNA extraction from banana. Every living thing has DNA to its cells, such as this one, the banana. DNA or deoxyribonucleic acid is a molecular composed of two polypeptide chains that coil around each other to form a double helix carrying a genetic instruction of the development, growth, and reproduction, functioning of all organisms as well as viruses. So today, we are going to extract DNA from the banana. So let's start. Right here in front of me are the different materials that we are going to use in extracting DNA from the banana. We are going to use the tablespoon, a knife, filter paper, funnel, salt, Ziploc, isopropyl alcohol, liquid detergent, the 3 sizes of beaker, the test tube, graduated cylinder, warm water, stirring rod, chopping board, and the most important material that we are going to use is the banana. So the first step that we are going to do is we peel the banana and we are going to chop it into a tiny pieces. So let's peel the banana gently. We're going to chop the banana into a tiny pieces. We are going to use banana with just the right amount in extracting the DNA. So after we chop the banana into tiny pieces, we are going to transfer it to the beaker and set aside. We are going to use a 200 ml beaker. After we chop the banana into tiny pieces, the next step that we are going to do is we need 50 ml of warm water and a teaspoon of salt. So this is the 50 ml beaker. The warm water. We need to put a 50 ml. And a teaspoon of salt. Using the stirring rod, we're going to stir the water with salt for about a minute until it blends it well. It is about a minute now, so the next step that we are going to do is we are going to put this one, the tiny pieces of banana and the salt water together using the Ziploc. I use the Ziploc as an alternative for the blender now the salt water into the ziploc. Let's smash it and blend it together. You need to do this one until you can see the banana is already smashed. As you can see, you already see the bubbles right here. So let's smash it more. As you can see, it's already smashed and blended well. Now the next step is we need to transfer this one to the beaker again. Using this one, the funnel. We need to do it slowly. The next thing we do is we need to put some liquid detergent, a teaspoon of liquid detergent into the sample. As you all know that every cell of the banana has a DNA inside, but it is wrapped up in this bubble called the plasma membrane. So we are going to put some liquid detergent on the sample. We are going to stir again using the stirring rod. So as I put a detergent on the sample, you can observe or you can see that the detergent, it serves to break down the molecules that make up the plasma membrane on the banana. So after we put detergent on the banana, we are going to stir this one for about 5 minutes. So let's just stir it gently, slowly. So as I stir this one, as you can observe, you can see already the plasma membrane of the banana cells that have been broken by the detergent, and it released some of the DNA inside. So let's just stir this one for about 3 minutes. But when you see here at the beaker, all the DNA of the banana is still mixed up to the other gut and bits from inside of the cell. And all the proteins, all the cell wall of the plasma membrane, as well as the carbohydrates and many more. So in other words, you cannot see clearly the DNA of the banana right now as we stir it. So what we are going to do after this one is to separate the banana's DNA. So we still need to stir this one for about a minute. 5 minutes, we managed to separate the denser component of the cell, which gathered down the bottom over there. And this less dense part of the cell, including the DNA, are now floating around, sort in the middle. This one, the bubble-like. We can now see clearly in our own eyes. Here, look at this one. This one are the DNA of the banana. The bubble-like floating on the beaker. This one. So you can now see really close with your own eyes the DNA of the banana. As we want to separate more to see the clearly DNA of the banana, we can use the filter paper. So as we want to separate more to see the clearly DNA of the banana, so we can use the filter paper, this one, to pour that into the graduated cylinder together with the funnel. So as we pour this one, we are getting rid of the larger chunks of the banana and some of the froth from the liquid detergent, which will get on the way of the next step. So now, we're going to filter it. Pour it slowly and gently. And wait until it filters all of the sample. So after 10 to 15 minutes, this is now the result. So the DNA is now dissolved in the solution, and using the isopropyl alcohol, we need to precipitate it to make the DNA more visible in our beaker. So I will transfer this one to our beaker. Observe this one. I will slowly add an alcohol on the solution. Really slowly, so you can now see the nice layer of the DNA forming on the top of the solution. So let's pour slowly. As you pour that alcohol down there, the layer forms if you observe it really closely. So there is a separation due to the isopropyl alcohol. When you see it really close, you can observe the boundary between the layer of the alcohol and the layer of the banana. Right here. You can start to see now the cloudy bubbles on the top of the sample, and it starts to form the what you call the DNA precipitate. So I will transfer it here so you can clearly see. The DNA precipitate of the banana. You can see clearly now with your two eyes the separation, the boundary between the layer of the isopropyl alcohol and the layer of the banana. You can now see the white cloudy bubble on the top of the sample that starts to form the what you call the DNA precipitate. This one. So that's how you extract a banana to see the DNA. So easy to prepare the materials and it's easy to do. I hope you enjoy seeing the banana's DNA with your own eyes. I hope you gather idea and knowledge through watching this video. I am Siamena Trubiana, third year biology student of Mariano Marcos State University. Thank you for watching.